the aims of that particular RFA. So with that being said, I think all the projects we're going to talk about are fantastic, and part of it is, like Will said, is not only meeting the criteria of the particular RFA, but making it abundantly clear. And because some of these things, like what exactly, how translational does it have to be translationally relevant, is a little bit in the eye of the beholder. And so if you don't specifically address that and make the case of why your particular research project has translational relevance, it may or may not, depending on sort of the eye of beholder or reviewer, be obvious. And so explicitly addressing, especially when you have a grant application that has really specific criteria of things that it's looking for, laying it out for the reviewers of make your argument and make your case like you're a lawyer of why your project is going to be relevant for translation in the future, really helps the reviewer check that yes box, rather than, as we'll talk about, if there's some looking through, you can see the reviewers, just some of the reviewers, it wasn't tight. They were actually not sure. They sort of wanted to believe it was translational, but you could see there's a couple of comments where they said it wasn't entirely clear. So if you can make that case explicitly, sometimes that'll help push your grant over the edge too. But just because you didn't fulfill the criteria for a particular RFA doesn't mean it's not a great project, as Will was saying.
So actually, that's a good transition to Michael, who I think wants to spend a couple minutes talking about this particular mechanism and the reason the criteria and the way it's crafted the way it is. So Michael, take it away. Any comments you have about NIH study section or this?
Before Michael,
Thank you.
Before Michael starts, I want to say that I just started recording, so all the critiques will be recorded, and I apologize I didn't start recording when Will was going through all those pearls of wisdom, so I apologize for that, but the rest of the session will be recorded.
Good. You didn't get me swearing on camera, so thank you.
I did not, but if there's anyone who's not comfortable with this being recorded, please let me know. All right, Michael, take it away.
So Will, Dan, Rose, thank you for your kindly introductions. I want to say for the group that Rose Viscardi is one of the most experienced reviewers and grant applicants I know. So if you have any questions, you shouldn't hesitate to discuss them with Rose. The way Dan and Will have laid things out is highly accurate picture of the way study sections run. In answer to Will's question, the study section review officer, the individual from the NHLBI in charge of the review, and the review chair will usually order the applications in the order of best priority scores, so that the first application the study section or the review group looks at is the one that the initial reviewers gave the best score on average. And then it goes down to, as Will said, about the 50% point, and the discussion of the applications stops. That means it's important to give your application the best possible chance of getting to be the first or second application reviewed, because that's when the reviewers are fresh and in a good mood and not cranky and not rushed, because the only changes in that order are made to accommodate somebody whose airplane or next meeting is coming up fairly soon, or and the ones at the end get less thorough attention and less sympathetic attention than the ones at the beginning. And that's relevant, even though the structure of the Jumpstart Award review is different. It's relevant because the reviewers don't discuss, this is all done remotely. The reviewers in the Jumpstart Award cycle reviews do not discuss their reviews with each other. So if you get 4 reviewers and 3 of them give you good to really great scores, and 1 gives you a terrible score, what happens is that all gets averaged up. The final average scores get sent to Denny Buxton, and then Denny looks over the applications, and if he thinks that the 3 reviewers who gave you 1s were spot on, and the reviewer who gave you an 8 didn't know what he was talking about or she was talking about, then he will slip you into the funded group if your average is within striking distance. But if Denny looks at the reviews and thinks that the reviewers who gave you all 1s were missing something, and the reviewer who was critical found a fatal flaw that would make your project not feasible, he'd rather see you resubmit. So give it your best shot, because that's what makes the reviewers in the Jumpstart Award review most likely to give you a score that'll end up getting you funded. In the Jumpstart Award reviews, you don't have the chance to have people who think the application is really strong correct the individual who's mistaken. It happens at a programmatic level when Denny looks at them, and there's a lot of deference to the independent reviewers so that Denny doesn't get accused of playing favorites. That way, make your application as clear and simple and as strong as possible. And when you send it in, if it's a resubmission, make sure that you address the point that was criticized on the initial submission so that it's now clear. And this applies for ordinary study section reviews as well as the Jumpstart Award reviews, that it's clear that that's a concern that you thought about and that you had a way to address it that doesn't leave you just twisting in the wind. So that's all I have to say, except stay in contact with Rose and me and Ling and Andrea and Liz and Dan and Will as much as you can. We'll do everything we can to help you.
All right. Thank you very much, Michael. And you actually triggered in my brain a piece of information that I had but forgot to say, which is that the other thing I know about order is that sometimes the program officer will alternate the beginning and the end of the priority score list. So they'll talk about a very high priority and then a very low priority and then a very high priority and then a very low priority. And I've had faculty who've served on a study section organized in that way say that it helped them understand the range of grants that were under discussion and appropriately set their expectations for what was a really good grant in this study section on this date, which I guess can be something of a moving target. Rose, before we move on to review the applications, do you have any pearls or anything else you want to add beyond what we've said?
Yeah, I have just 2 pearls. One is more for applying for either to a specific RFA for NIH. Is usually these RFAs are written by the program officers whose names are listed on that RFA. It's their baby. And so one of the things that you can do is call them ahead of time before your submission deadline and tell them about your project. One, to see whether they think it fits their RFA. And then they're usually very helpful in how you could craft your aims. They may even ask you to send them your specific aim page so they can give you some feedback. And that can be really helpful in being sure that you are responding to that RFA appropriately, and they can give you some tips on how you might be able to reframe your aims to be able to do that. And then the program officer can be helpful at the other end as well. Once you get your scores and you've gone through the stages of grief, as Will described, you can arrange to speak to the program officer, and they can help you kind of read between the lines and figure out what you need to do to address the main concerns of the reviewers and submit a competitive resubmission. So the program officers can be very helpful both in planning your application, and as well as, if need be, planning what you need to address for a resubmission. So they can be very helpful, as well as having other colleagues look at the comments, because sometimes it's hard to be objective, as Will was describing. And so sometimes getting other colleagues to also look at the comments and your application to say, you know, they're right about this particular aspect of the application, and this is really a weakness that you need to address. So that can be very helpful. So get all the help you can is where I'm going. All right, well, do you want to start the first one? We have about a half hour allocated for each one.
Yeah, I mean, I think we're on track here. We'll start with Christopher Thom.
Thom, nice.
Thom. All right, Chris.
Good.
So who was our reviewer for this one besides me and Dan?
Is it me?
I don't think I ever got any sort of assignments, but I did all three. So.
All right.
All right. Well, you just volunteered.
I was going to say you're now our backup. So
Great.
the call
Great.
just you're running the show.
Wow.
I was, you know, as Will said, though, it's actually a good point, right? So there's a primary reviewer, but the primary reviewer is the one who's going to, you know, take the lead. And so there's other reviewers, but they're also reviewing a lot of applications. And so, you know, that may sometimes not be super far off from reality, where the primary reviewer is the one who spent the most energy, and there's other people who have reviewed it. But, you know, as I'm sure you've heard, you know, reviewing grants is one of a million things that senior faculty do. And so sometimes this happens late at night, or like on the airplane, things like that. So, you know, even though you have highly experienced, really smart people reviewing, sometimes they're doing it really tired and distracted. And so, like Will said, that's, you know, sort of emphasizes why it's important to just make it as obvious as possible that you're meeting all the criteria in the grant, because sometimes people are reviewing a million grants and are distracted by other things. So that, you know, maybe that's a good example of reality. So I am going to serve as a primary reviewer for Chris's grant. So I'll just start by summarizing it. And then what I'll do, since this is our first grant that we're going through for this study section, I'm going to actually go through and read the review guidelines for each criteria before I give my assessment for this particular grant. And then I'll give the overall impact score and go through the criteria and what make up the overall impact score. And then we can have a discussion, and we could, Will, I don't know how you want to do, but we could talk about the reviews that the grant got as well, and how that fits in with what we thought. And then we can do our secondary reviews. So just to start, so the title of Chris's grant is Defining Mechanisms by which Tropomyosin-1 regulates in vitro hematopoiesis. And the question that they're trying to address in this grant is that obviously, for a variety of reasons, producing red blood cells in the lab for therapeutic use is obviously an attractive goal. There's a lot of reasons why there's a shortage of donor red blood cells, or that the patients have a variety of immune considerations that make it hard for us to match the appropriate red blood cells that we might have available from donors. And so producing red blood cells in the lab for specific indications would be a very valuable option to have in the clinic. But the biggest limiting factor in achieving this goal has been the production of an appropriate volume of red blood cells. So we'd need a huge amount of red blood cells to actually transfuse to a patient. So we'd really have to be able to produce them at massive scale, which has been a huge limitation of the field. Chris and his group has identified that deletion in trypomyosin-1 gene results in increased hematopoietic progenitor cell production of red blood cells. And the purpose of the grant is to understand the molecular mechanisms by which a lack of trypomyosin-1 leads to increased red blood cell production, which by understanding the mechanisms could potentially be applied therapeutically to increase red blood cell yield for use in the clinic. And the authors, this is based on, they have good preliminary data that they present in their grant suggesting that deletion of trypomyosin-1 results in increased red blood cell production. And it also looks, by looking at his CV, that they have a paper that's been submitted to bioarchives, which means likely it's in review at a journal currently. And so he not only has good preliminary data, but it's also the basis of publications that are directly relevant to this grant, which speaks to his strength as an investigator and their ability to accomplish this project. So that, I think, and then the way in which they're going to tackle this problem, they know. So tropomyosin 1 is related to the actin cytoskeleton within cells, and this is directly related. Changes in the actin cytoskeleton are known to directly be related to a modulation of key signaling pathways like Wnt and Notch, as well as Hedgehog. And so they're proposing that by deleting tropomyosin 1, there's alterations in either Hedgehog, Notch, or Wnt signaling pathways that's contributing to increased red blood cell production. And to address that, essentially what they're proposing to do is perform RNA-seq, bulk RNA-seq at 4 different points of differentiation of the red blood cell precursors to look at the signaling pathways that might be altered as a way of understanding and potentially manipulating this for therapeutic gain in the future. So just to go through the criteria for this specific RFA, and I'm going to read through. So the guidelines that are given to the reviewers, the first one on the list, which speaks to the translational nature of this grant and the PCTC in general, is they want the reviewers to address the relevance to translation. So the reviewers are specifically told to spell out briefly how the work proposed will advance the translation of progenitor cell. Screen keeps on popping up. Let me go back to my. Will advance translation of progenitor cell basic science and bench findings to application for diagnosis or treatment of humans. The advance could be at an early phase, i.e. from in vitro to small animal model, or from small animal model to large animal model, or a late phase from large animal models to good manufacturing practices necessary for the production at scale for first in human administration, but must move the state of the art forward from one point somewhere on the spectrum of progenitor cell discovery to application as a diagnosis or therapeutic in humans. And for this particular application, I would say this is obviously on the basic science end of the spectrum. We're not translating into even an animal model of disease, let alone kind of directly to human human studies. But I think what's clearly relevant is Chris has nicely laid out the problem is we want to produce red blood cells for use in the clinic, and we're not able to. We can make red blood cells, but nowhere near the volume that we need to make for therapeutic use. He's identified through his genetic studies genetic variation that leads to increased red blood cell production. And by understanding the mechanisms, you could particularly either genetically manipulate cells or pharmacologically manipulate cells during iPSC differentiation that could produce increased red blood cell production in the future. So it is at the basic science level, but I think there is obvious translation down the road if Chris were to successfully achieve his project and understand why deletion of this particular gene leads to increased red blood cell production. The next criteria is application of a new technology to an existing translational research question. Technologies that have used and are well known are of less interest than newly developed technologies that could become game changers in the way of overcoming obstacles to translational research with progenitor cells. Novelty of a technology alone is not enough. It must be novel, a potential game changer, and applicable to translational progenitor cell research to merit a yes. And as we'll talk about as we go through the other reviewers that have reviewed this grant previously, this is a really high bar. And so like we talked about, just because a project is great doesn't mean it's going to necessarily meet all the criteria that the RFA has set forward. And so for this particular case, I'd probably agree with some of the other reviewers in that application of RNA-seq is potentially going to show us some exciting things and really kind of open up an area that's potentially translational and understanding the mechanisms that are really leading to increased red blood cell production. But I would say no, there's not a new application of a technology to an existing translational research question. The next question is preliminary data for new translational research question. Jumpstart projects that can be anticipated to produce preliminary data for new translational research questions that could form the basis of future investigations or funded research are valuable to the investigator and to the PCTC. If the output of the Jumpstart Award project is going to be data that could be preliminary data in an investigator-initiated grant with a progenitor cell translational research specific aim, i.e. advance the translation of progenitor cell basic science bench findings to application for diagnosis or treatment of humans, the advance could be at an early phase, and that's using the in vitro to small animal model or a large animal model to human clinical trials. Move the state of the art forward from one point somewhere on the spectrum of progenitor cell discovery to application as a diagnostic or therapeutic advance in humans, it merits a yes. And in this case, I would absolutely say yes for this project. This project is, as Chris outlines in his application, potentially generating data, understanding molecular mechanisms by which tropomyosin deletion results in increased red blood cell production, which has obvious therapeutic implications. And it'll clearly, if successful, generate preliminary data that he, either the PCTC or his PI, or hopefully him himself, can use for preliminary data for something like a KO8 application or other early career stage investigator grants. So I'd absolutely say yes. Establish and the next criteria is establish or prepare a new inter-hub collaboration. This criterion merits a yes if the Jumpstart Award project will involve work by a postdoctoral fellow or investigators in laboratories in more than one PCTC research hub, and those laboratories have not previously collaborated on progenitor cell translational research. I think it sounds like this will all be done in Dr. Tham's current lab, and so I don't think that there's probably an inter-hub collaboration, but as we'll talk about in the criteria, this isn't exactly a deal breaker. So I would say no for that, but we can get into how that assesses the overall kind of assessment of the grant. The next criteria is objectives are clearly stated. Approach is reasonable to achieve this objective within the described budget. And then it lists some criteria. Again, Chris proposes, I think, a very doable set of experiments, both given his experience and qualifications as well as those of his mentors. He's proposing to do RNA-seq at 4 different time points on iPSCs during differentiation. I think this is absolutely doable within the budget and within the training and experience that he has, as well as the environment that he has at CHOP and Penn. So I'd give this a yes. And then we get to our overall impact score. So the overall impact score that we'll talk about a little bit more is based on several criteria. And it's not necessarily a direct sum. So reviewers will usually give a score on each of the criteria. So significance is obviously the significance of the research question and the overall grant. Then there's evaluation of the investigator. So whether the investigator is qualified and trained to perform the work that's being proposed. Next, the grant's evaluated on innovation, and then evaluated on approach, and then finally the environment, which is obviously the greater lab environment and the institution that the work is being provided. So, for example, does the investigator have all the resources and core facilities that they need to complete the proposed work? And that might include things like a specialized equipment that might be necessary, core support like bioinformaticians to do the data analysis and that sort of thing. And so reviewers will give a score for each one of those 5 categories, and then an overall impact score. The overall impact score isn't necessarily just an average of those things, because obviously you could have a great investigator and a great environment, but if the question's not good, just because you have a fantastic investigator environment, if the question's bad, then the overall impact score is not necessarily going to reflect the fact that is simply an average of the fact that the investigator's great, but the question is bad. So it's not just you add up those 5 scores and take an average. And so for this particular grant, I would say, so to go through those criteria for significance, I would give this application a 2. And remember, this is on a score of 1 to 10. So I think, as we talked about, particularly in the context of this RFA, that the application is likely to provide important biological insights into understanding how tropomyosin 1 deletion leads to increased red blood cell production, and that it's indirect, but has down the road clearly has translational implications if it leads to methods that we could use to increase red blood cell production during iPSC differentiation. The next criteria that we evaluate is the investigators. Here I would give a 1 for Chris. He's had an outstanding training record. He's an MD PhD with, as I mentioned, a number of publications that looks like currently either under review or in press, as he's submitted work directly relevant to this grant application that's been already published on bioRxives. And then he has a long-standing and very productive record throughout his training as both a PhD and during his fellowship postdoc period of being productive and publishing in high-impact journals. So again, I give his investigator score 1. Innovation for the project, I give a 3. I think this work is going to lead to exciting biological discoveries. The limitations here, I think, are that the application bulk RNA-seq is a very powerful but technology that's been around for some period of time. And so a series of, RNA-seq experiments, I think, is going to generate some exciting data, but it's going to be more in the category of preliminary data. I think for the context of a Jumpstart Award, that's totally fine, because the purpose of this, in my mind, for Chris, is to generate preliminary data that he can use for a K-weight application. But, I'd say I'd reserve a higher innovation score for, more cutting edge or, sort of out of the box technologies, which again, in the context of a relatively short duration grant that's targeted at investigators, I think is a really high bar to achieve. The approach, again, for similar reasons, I'd give the approach a 3. I think that this is a very solid approach and is going to generate data that's going to be very useful and generate preliminary data, both for a grant, but also that can be directly follow up with additional targeted studies that will directly answer the questions that Chris proposed. But again, I'd reserve kind of the highest scores for, a really kind of, really out of the box kind of cutting edge technologies and, so yeah. And then in terms of environment, I'd give this grant a 1 in that category. Chris has both relating back to the investigator, but certainly the environment he's got fantastic mentoring team in a fantastic environment at CHOP and Penn. I have no doubt that he complete the experiments that he's proposed in this grant. So putting all that together, again, remembering that this is not an average of those scores, I give an overall impact score of 2 for this grant. And then, so that's my review. Will, do you want to have the other people present their reviews, including you, or do you want to go over the reviews that we have from PCTC?
I was going to say, why don't you give us a bottom line score, and then we'll ask Jessie to act as the second reviewer, give us her score and what comments she has to add to yours.
Sure. Yeah, that sounds perfect.
And then I'll go third, and then we'll bring in the prior reviews and see if we can synthesize some advice for Dr. Thom.
Yep. So my overall impact score was a 2.
Okay. Jessie, what score did you give Chris?
My overall impact score was a 4.
Okay. Go ahead and tell us, justify your score and add whatever you have to what Dan said about the grant.
Okay, I think most of my comments echo what Dan said. I thought that the relevance to translation was very high due to the potential use of iPSC-derived red blood cells as a source for clinical blood reagents. So translation was high. The application of a new technology to existing translational research was, as Dan said, lower on the scoring because there were no novel technologies listed. Again, it's just bulk-seq along with the CRISPR-edited triple myosin 1 knockout lines that the PI already has. So unfortunately, I gave that a higher score. The preliminary data that was shown in the grant was very relevant, and I think was a strength to this application and shows the feasibility of some of the work that has been done. And I agree that the discovery of these mechanisms can definitely provide some preliminary data for future grants downstream. As Dan said, there was no evidence for new inter-hub collaborations, so that was not a part of this grant. For objectives, I think those were clearly stated. The justification of the approach was very well laid out. I thought that sample collections and requirements all were fair and fell in the described budget. One of the limitations that I saw was that what was listed in the anticipated results was identifying novel targets, and I thought that might have been a weakness due to potential of not being able to determine those novel targets, and maybe the same pathways such as Wnt and Notch as the PI has listed will come up. So that was, I guess, one of the comments I had, other than what Dan had said. So I think I agree with mostly of what Dan said, and those were my comments.
All right, so I'm not going to add much in terms of the critique of the grant. I think there's an argument to be made that the answer to is there an application of new technology to an existing translational question could be yes, if the argument was made more strongly in the grant that the differentiation protocols that are under investigation with this study are truly novel and cutting edge. I don't know if that's true, but I think the argument that those are cutting edge techniques did not come through. You're not going to have an argument that's going to make bulk RNA-seq sound interesting. Ironically, the grant would score a yes on this if you decided to do, say, single-cell proteomics, whereas bulk RNA-seq is far more likely to answer the question that you actually have. So this is one of those yin and yang things where you have to try to propose the technique that matches the question in order for the approach to be reasonable. But you also have to try to give it a little whiz bang. So the argument that I would make around this is that this is a relatively interesting, yes, Chris, interesting technique, and that you could do that you know your cell line is novel, it's engineered from a particular history, and that cell line is the best possible technology available currently to answer the question that you have, might push a couple people into the yes category for that question. The last issue that I'll mention is some of the discussion about can you do all of the work in the budget that you proposed? And this is going to come up on all of the grants, actually, I think. The challenge with a $25,000 grant that is labeled a Jumpstart Award is that you have the impression that you're supposed to use it to do the first part of something. And that's true. But you can't make it sound like you're doing the first part of something. You have to try to encapsulate the work that you're doing in such a way that it sounds like a relatively complete answer. So I think that in this case, is this approach reasonable to achieve the objective within the described budget? I would say yes to this grant, because I think the grant is well circumscribed around the RNA-seq experiment. And I think that the justification for the RNA-seq experiment is strong, and it will answer the question that's raised by the investigator in their specific aims. However, I can see someone saying no to that based on the fact that all you're going to get, as Jesse said, is a list of targets, which tells you nothing intrinsically about actionable biology. You might get super cool actionable biology out of that. You might not. You don't know yet. That's okay. You have to do the experiment to find out the experiment is justified, but to try to make the argument that you're going to get an answer that's interesting regardless. So some sort of language like this appears to be a key transcriptional regulatory time point. At minimum, we will define a transcriptional regulation downstream of blank molecule, blank pathway. And we hope to be able to tie this into signaling pathways. Might make that argument a little bit more linear and strong. So I like the grant the best. I gave it a 1. And the reason I gave it a 1 is because I think that of the grants under consideration today, this is the most molecularly mechanistic grant. It's got the best preliminary data, and it's got a fundamental question that I think is likely to be translationally relevant immediately. Whereas the other grants that we're reviewing today, and some of the other grants that I reviewed in prior for this cycle, were technology development. So I like the idea that this is a molecular pathway and a defined question. And I think that really strengthens the grant. I frankly like the fact that RNA sequencing was chosen as the technology because it's the right technology to answer the question that Chris is trying to ask. It's focused on, he has questions about subtle pathways that will not be well described by a single cell RNA sequencing experiment. And I think it's the right answer to do a bulk seq in this case to get to get his good question answered. And so for all of those reasons, I think this is a high priority project. But I think that the fact that I'm giving it a 1 does not take away from, I think, Jesse's reasonable critique, which is that with this sort of an experiment, you don't know what the answer will be. And I think the grant could be strengthened by trying to define a little bit more what the answers you'll definitely get are versus the ones you hope to get.
Yeah, and I think, you know, some of the discussions we're having clearly show some of the trade-offs when you have, like I said, really high bar criteria that you're trying to meet where, you know, as in some of the chats that are popping up, you know, you could say, well, what if I try to increase the technological excitement, so to speak, by doing single cell RNA-seq or single cell RNA-seq and ATAC-seq, because that's the new and coolest thing that 10x is selling right now. And you can see how, you know, as Will pointed out, that you're probably going to get the answer to your question with an older, quote unquote, less exciting technology. You're probably going to get a better answer to your question that's going to lead you to more definitive downstream experiments with something that's maybe not as the kind of thing that's in vogue right now. You know, whereas if you take the newest and greatest technology and check off that, you know, exciting technology box, quickly you're going to take yourself out of budget and maybe make it less clear of how you're actually answering your question. So there's all sorts of trade-offs, and it's probably impossible to make a yes to everything. The other argument I'd make is what Will has taught me over the years is a lot of this is advertising and salesmanship is as much it is that, you know, actually, you know, the project. And so making it clear, as Will pointed out, some of these categories, you know, whether it's translation is a little bit in the eye of the beholder. And so making a clear case in your grant of as Will said, you know, it's not like everyone is making red blood cells in their garage. And so the fact that you have this really exciting technology where you're making red blood cells, yes, you're using bulk RNA-seq, which is not a brand new technology, but there's not that many people doing this. And so you're using really exciting cutting edge technology, even if the, you know, actual experiment involves bulk RNA-seq, which is something a lot of people do. So just making that case clear in your grant can make, you know, that case for, to get some of the technology boxes off that if you leave reviewers to their own devices and try to figure it out, it might not be quite as clear.
Can I have one comment? I don't know if you guys allow other people to speak.
No, of course. No, this is a discussion, Sarita.
I know. I just don't know if there was a time. It's funny. If we wait 10 more years, bulk RNA-seq will be the coolest thing because nobody will be doing it. I think one.
Yeah, it'd be like vinyl records.
Right. Yeah. Exactly. I think the one part of it that I you know, as a reviewer would have been really nice to understand and/or get into the mindset is why transcriptional regulation is the thing that we think is happening. And I think Will maybe alluded to that already. Maybe there might be some more proteomic approaches that could be used, and that would have been the cool part, perhaps combining like an early transcriptional profiling, like day 0-2, plus the proteomics later on, because I didn't get the sense of, you know, why transcriptional regulation is the thing that Chris thinks is the way this works. And I actually would argue that perhaps the from the protein Western that I saw, it's like such a, you know, the protein is there and suddenly goes away in the HSPCs. It seems like a less transcriptional and more post-translational or perhaps regulation that is in place. So I think that part might have been a little bit better explained, and that would have made the approach appropriate. And or perhaps as an alternative, instead of saying, oh, if it's not transcriptional, I'll think about like some other, you know, actin mediated ways that this can do it. It could have been like a proteomic approach as an alternative way. So that was my only thought.
Yeah, this is Rose. I would second that. I think all three applications could have been strengthened by having a section on potential problems and alternative approaches. So it would have been clear to reviewers that you didn't just expect that it was going to work out as planned. Almost nothing ever does. And it's really helpful to reviewers to know that you're thinking about what could go wrong or some roadblocks that you might run into, and then how you're going to approach that, particularly if, as you say, if you're kind of going down the wrong path to begin with, and you find that out, what are you going to do? How are you going to sort of refocus? So being able to both address technological potential problems with what would be an appropriate alternative approach, as well as, you know, if the work is not supporting your hypothesis, where else are you, you know, what else could you, if it isn't going as anticipated, if the results are different than you anticipated, then how are you going to address that? This is a very short award, but when you have a, you know, 4 to 5 year R01, often you actually end up having to revise your aims along the way because of the results that you are obtaining. So being able to think ahead of time of what you might need to do, what those pitfalls might be and how you might approach it, can give some confidence to the reviewers that you're aware that there's pitfalls and what they might be, particularly with the technology that you're using. And that you have some ideas of how you could approach the problem by a different approach.
And to add on to what Rose and Serene just said, it is a very short grant, but remember that there's that second page for justification of approaches in the budget. So something like what Serene had mentioned about the, you know, rationale for the transcriptomics can, I mean, I would have used that as well to kind of use that second page.
Yeah, I think that's an incredibly good point. I think that all three of these grants underutilize the budget justification as extra space to generate additional argument in these grants about what, not just like that the budget is what it is and you're going to spend X amount of money, but instead to say you know, you could almost in a grant like this say as an alternative we would consider blank, which would cost blank. And so if the first step doesn't work, we will transition to use this other technology. And that actually would be a very reasonable thing to do in your budget justification. And I think it's something for all three of you guys to think about moving forward.
And something else in terms of, sorry, one more thing. Something else in terms of
for Jesse, and then we're going to move on.
Something else for I guess what Will had said earlier about packaging and marketing the grant is also the inter-hub collaboration. I know it's a very hard section, but I would advise looking through the hub PIs and the projects and look for any potential, you know, collaborations that you could actually write up. It's not something that you have to carry out, but I think it would strengthen the grant a lot to have something about a collaboration.
Yeah, the inter-hub collaborations are getting harder every year because each Jumpstart Award is generating new inter-hub collaborations. So, for example, Jamin's project is a collaboration between two of the lung groups so now no one else can use that exact same argument because the two groups are now collaborating. But there is because the way the criteria are written, it's a new collaboration that has not yet been done. So I think that all three of you didn't even make noise towards an interhub collaboration, which I understand because the projects are standalone projects and they don't necessarily have to be that way. But I think thinking of ways where you can say that it's strengthening or building on an interhub communication or collaboration at minimum would really strengthen each of the grants. Michael, I see you popped in, so you may disagree that it has to be.
No, I want to say that though the lung people are working with each other for the most part, the blood people aren't. And Chris, if you could get somebody at Boston who knew about cytoskeletons to agree to be an advisor for a nominal sum, and you might draw somebody's interest too. I know Serene knows Boston better than I do, but there are other blood groups, and crosstalk among the blood groups would be very attractive.
I think I misunderstood then. 'Cause I thought it was like the blood hub. You know, like I thought it was going to the lung group was the inter-hub communication or collaboration.
Yeah, so that's interesting. No, it's just with any other. It's just with any other
research hub. There are 7 research hubs in the PCTC. There's the one in Boston, Daly, the one in San Francisco, Chapman, the one in Cincinnati, Whitsitt, the one in Alabama, Zhang, the one in Philadelphia, Chow, the help me if my memory is slipping, Rose.
Did you do Columbia?
The one in New York, Schnook, and the one in Wisconsin, Thompson, and a collaboration with any of those other research hubs would add a favorable sheen to your application. And it doesn't have to be somebody's doing the lab work. It can be somebody's supplying a model, somebody's characterizing your cells separately, somebody's advising you about cytoskeletons.
Yeah, I mean, even data for your particular project, even a cytoskeletal expert who agrees to help evaluate the data from either transcriptomics or proteomics with you to help understand that would be sufficient for me to say this is probably a new collaboration, because that's a communication that wouldn't otherwise be happening without your grant.
Thanks very much.
Dan, do you have anything you wanted to say from the prior scores, or did we cover it? You're muted. I see your mouth moving, but I can't hear anything.
Okay, sorry, I had it muted. So yeah, I think we covered pretty much everything that I saw in the prior evaluations. The only thing of note for everyone listening in is that you go through the 4 reviews. 3 of the 4 were very consistent, and then which all gave the application pretty consistent with what we're saying, a 1 or a 2. And then there was a reviewer who gave the grant 7. And it really wasn't entirely clear from my review that those comments are pretty thin. It just, the only weaknesses they stated on the review that gave it a 7 were uncertain if there will be a result. The plan is to simply compare cell populations. Also, there will be no evaluation of the quality of cell population. So you can see the variation even amongst highly experienced reviewers is 3 out of 4 gave it a really great score and thought it was a great application, and 1 person obviously really didn't like it. So that's always the challenge with getting your grant reviewed. And I don't know if you have other thoughts about that, Will, but that's just the way it goes sometimes.
I usually, when there's one really divergent reviewer, I tend to think that doesn't mean the reviewer's dumb, but that I didn't make the best argument available because I only captured 3 of my 4 audience.
I totally agree. And from his comments, you can kind of tell that he just wasn't convinced by the excitement that the other 3 people got. So I think it's more a matter of the project's great, and it's probably just a matter of spicing up the application a little bit, so to speak, on the second try.
Life is sales. Okay. Both science and medicine have huge amounts of sales in them. I'm always trying to convince people of this. Especially as a doctor, God knows we're not selling masks very well.
That's
So,
for sure.
all right. So, Chris, other questions for us, or comments, and then we'll move on to Anna's application.
Yeah, no, this was perfect. I mean, actually, I am sort of glad that Dan just said that, because that was my impression too, is that I just needed to sort of sell it more. So I think I very much appreciate your comments and ways to sort of reframe this for September.
All right. Okay, so I am gonna. Who's joining me on Anna's application? Was that Serene? Is that you? All right. Well, Jesse did the last.
So again, I'm happy to
Jesse
comment on
presented the last one, so you're up. I know Jamin's doing the heart one because I got emails from him about that one.
All right.
So, so it looks like you guys all reviewed all three. So please make comments. Like, please jump in and make comments with us on all three of the grants. Okay. But I will start by being the initial presenter for Anna's grant. So this is a very interesting grant describing the use of a new, much wanted technology, which is a pig ES cell, to be able to begin to derive porcine-specific endoderm and lung lineages through a differentiation of these ES cells. It has been a major challenge for many years to get any sort of ES or IPS cell from pigs and sheep. And so the fact that there is now an available line is an important advance. And so in terms of application of a new technology to existing translational research question, I would answer clearly yes. This is a novel technique. They are going to use protocols that are well established in the Chinook lab for differentiation of other types of endoderm to this novel cell line, which is an important potential translational asset. The most important aspect of the investigator here and environment is I think that Anna is well prepared to carry out these studies, and she's in an ideal environment to take on a difficult progenitor cell project. This is a lab with high throughput of different types of progenitor cell projects, established protocols for a number of different aspects of this project, and the capacity to be able to do this carefully with or without the money derived from this grant, which I think is important. This is something that I'm sure can happen in this environment, whether we give her this grant or not. In addition, I think that this is a really interesting preliminary data set that she's proposing to generate. So I would say that the proposal does generate preliminary data for a new translational research question.
We lost Will.
I
Yep.
think he's frozen.
I don't know if he knows he's frozen.
I don't know. Oh,
I'm back.
yeah, you're back.
Okay.
All right. I heard the last thing I heard was Serene say, "I can't hear Will," and then so just to go back, I'm just going to say it's a little bit unclear to me exactly what Anna would propose to do translationally with these cells once she got them. The suggestion is in one sentence that perhaps they could be engrafted into bleomycin mice, which I think is a divergent question of whether people think that's important or not. My personal bias is that bleomycin mice are not a particularly good translational model, but that some more argument could be made around that would define it a little bit better for me. In terms of objective clearly stated and approach reasonable, yes, I think to begin with, the initial characterization of the cells is well described. I think the process makes sense. And Anna did spend some time in generating some additional discussion of what markers she would look at. Like we discussed in the prior application, I'd like to see more here in relationship to what if the markers in pig are a little different than the markers in other tissues. We know that mouse and human markers are a little different. We know the differentiation protocols are a little different. Can we think about how pig might be a little bit different from those as well? Does the proposal establish or prepare for a new intra-hub collaboration? I would say no, though I think there is a significant possibility to work on this moving forward with the grant. I would, in general, I gave this grant 3. My major strengths are that this is I think this is an accomplishable project. I think it's well defined, and I think it answers an important translational question. The weaknesses of the grant are it's not totally clear to me how a pig cell is more helpful than a human cell for a translational question. It's also not completely clear to me what will be done with the cells once they're obtained. And then my last weakness is that there doesn't seem to be a driving hypothesis that underlies this other than these cells would be useful. And so it feels like a technology development grant to me. I'd like to see more in terms of details about how the cells would be used to answer important molecular questions beyond simply as a tool. And so for those reasons, I gave it a 3. But I like the grant. And in the recommendation for approval, I would say yes. But I think that some of the weaknesses could potentially be addressed on a resubmission without much difficulty that would strengthen the grant quite a bit. All right, so Serene, I think you're number 2.
On? Yeah, cool, thank you. So yeah, I definitely like the grant as well. My overall score was 2 for this. And the points taken really were about the approach and some of the details that I think Will already mentioned, but I would add the details of the in vitro culture, I think might be, I actually thought like what cytokines would you use, for example, mouse or human? Like, is there a pig cytokine panel that you're gonna have, and why do you think that those will work in pigs? So that was one of the issues that I thought would be interesting. It's actually interesting, Will, that so Hans's letter has the reason for this cells to be differentiated because Anna is working on making a model of porcine IPF, right? Is that true, Anna? Yeah. It's something like in the process of generating a swine model of IPF by deleting the HPS1 gene. So it sounds like there's some work that is actually being done in a large animal to actually create the models where these cells will be useful. So I think that, just to second on Will's point, just make a very big deal out of that, because that's really...
So I'm glad you pointed that out, Serena.
I would not be reading that letter, right?
I specifically ignored that.
Yes.
Because Anna didn't make that argument. So I wasn't sure if that was what she wanted to do. And we all know, right, in these letters of recommendation from senior PIs that they say what they're thinking instead of necessarily what you are. And so I didn't know what to do with that piece of information and specifically ignored it in my evaluation of the grant. So I think your point is extremely on point. That's a key thing that needs to be harmonized.
Yeah, and I totally agree. I think I'll talk about my assessment in a little bit once Serene finishes, but it was interesting because when I first looked at the title and the abstract, I thought exactly what Will said. It was like, well, why don't you just study human, because we care about human diseases. And then as I read the full application, I was like, oh, that's really cool. It's a great point. Mouse, for as Will alluded to, there's a lot of reasons why the mouse models we use, like bleomycin, probably aren't great for relevance to human disease. And they may, the point of large animal models for translation for human disease and understanding how we're going to do progenitor cell therapies is a great point in an important area before we're going to get to human translation. And so the argument after I got the whole grant is that, developing pig iPSCs is actually really important because it's a great intermediate step to translation. And so, but, I had to, think about it and read the grant more carefully before I got to that level of excitement. So I think that again speaks to, I think the project and the idea behind it is fantastic, but I think by the variation that you've just seen amongst even the three of us in a couple minutes about that level of interpretation, it shows that it probably needs to be made a little bit more clearly, so that the excitement is there right away and not after you carefully go through everything.
It should be like in an abstract or something, like
Exactly.
model is in process, these cells are going to directly feed into it, kind of thing. And the only last thing I'll add, and I don't know, maybe I'm just OCD a little bit about these things, is like there's a lot of little typos in the text. And I think just generally speaking, I have had that feedback a lot myself, like, and I'm like really big on that. And now is to pay attention to those little things, because reading the sentence twice makes me realize that, oh, it's not an N and it's an N, and then that makes sense, and then that makes the reviewer kind of be like, oh, come on. And so just to make their life as easy as possible, because Will said they're reading this at 5 a.m. on a plane, sleepless, etc. And just attitude changes, that's all.
And I think, Anna, in your reviews from the formal process, I think one or two of the reviewers pointed out the typos as well. And so it's certain people pick them up in a way that is very, it can detract from a really good grant, even a single weird typo, particularly if it comes in an important place. For example, God help you if you have a typo on your specific aims page for an R01, because everyone on the whole panel is looking at it at the moment they're scoring your grant, right? So that should be a never event, because enough people should have looked at your grant by the time it goes out that should never happen. But it just emphasizes that it's really important to remind yourself about that.
Yeah. I mean, and some of them are not like typos, but they're actually like scientific, like genetically versus generally, like things like that. And then like a couple of times I saw like a different reference to the same comment that you had. So obviously it's like a complete technical thing. It's just, just read that one more time and do everything, make other people read it. I think you will always catch these kinds of things.
All right, Dan, what was your score? And do you have any more comments?
So yeah, so I gave it an overall score of 4, and it's interesting because I thought more or less the same thing. So I don't have a ton of new things to add. I would say, so I think my overall level of excitement for what's going on is actually really high. I think especially, and again, as we just talked about, it took me a little bit to get there, because my initial reaction is, well, why would you make pig? Because we're already making human. But once I realized why you're doing that, I thought that's actually really exciting and a great idea and something the field needs to do. So I'm totally sold on the project and what's going on. I think the limitations, particularly from a grants standpoint, is more what Will said, which is the biggest challenge is that it's really a technology development. You're trying to differentiate these pig iPSC into anterior foregut, endoderm, and eventually lung. And so it's more of developing a technology. And so the thing is, there's technical questions to be raised of how you're going to really do that, which is hard to convey in a relatively short grant application. The other limitations from a grant standpoint, as Will said, and it depends on the nature of the grant. This is more of a translational grant, but there's not really a true hypothesis and research question that you're answering that you're going to be able to answer yes or no. And so tied into that, because it's technology without sort of a direct research hypothesis, it's very possible that you try a bunch of different cytokines or things and it doesn't work. And then you don't really have an answer to a question. You just have, an inability to achieve a technological goal that you set out to achieve. It's almost more of a bioengineering problem or question. And so it's very possible that you could do this and kind of get stuck in a rut, and you'll get information, but maybe not in the same way as if there was a specific research hypothesis that you're trying to answer. So I think that's a potential limitation, and certainly something that I think reviewers are going to pick up on. That being said, the project and the technology is really exciting.
And I suspect that you will learn a lot by failing in this project if you were to fail.
Yep. And so again,
tell us what that is.
Exactly.
So then I can say, oh, this is the academic product if she can't make the cells.
Right. And I totally agree with Will. I 100% think that this project is exciting and worth doing. It's just that on a study section, the naysayers are going to be like, well, what if you just don't make anything, and then you've spent all this PCTC money and funding and you haven't got anything. I personally totally disagree with that. I think you're going to learn, even if you don't, I think you're going to learn a ton of really interesting things, potentially about interspecies differences that'll be really exciting. But you have to sell that and make the reviewers think that, because if they're grumpy, they're just going to say, you're just trying to do something, it might not work, and then you won't have anything. So I'm totally sold, but I, as a sort of almost to help the kind of argument and study section, that's why I scored the way I did, because I think that's what some reviewers are going to say, is that if it's a technology question and you fail at achieving your technological goal, you're stuck, which I don't think is true. But
Jamin, I see you. You
Yeah.
ready?
Yeah, I have a quick comment to add. I think I was not the primary or secondary reviewer, for the matter, not a tertiary, right? So coming from a person who would not necessarily read your grant in the ultimate details and then advocate for the grant, I think one key thing that was missing for me is that whether in the grant itself, there was no explicit mention of whether the applicant has experience with the EPSCs and where are they going to get them from. The references that were cited were not from the lab. So do these cells already exist in the lab? Do the applicant have any experience with that? I think that's a pretty critical feature, especially knowing that progenitor cell or ESCs or iPSCs are really finicky to work with. Every lab to lab, they're going to differ. So that's pretty critical. And second, I second Serene's comment about the question that is there, the more details are required, like what cocktails you're going to use to induce differentiation, at what time points. And piggybacking on Dan's point, it's pretty, I think, also coming from the lung field, it's fairly easy to kind of make a biological question out of the grant that already exists, which is to, I understand the shortcomings of a bleomycin model, but considering it's an NSG mouse, you don't really have anything else. You can't use flu or any other viral injuries. So you could pretty easily just make a biological question that porcine-derived lung progenitor cells, are they as good in a bleomycin model as a human-derived lung progenitor cells? And that's a simple thing. And then you can come up with a technology to answer the question, which is the derivation of these cells.
Yeah.
Just my 3 comments that I have.
I like all of this. I think those are really on point. The one piece of advice I will give you from my own experience is that you have way too much background on this grant. I mean, like way too much. You need about a third of as much background and about 3 times as much detail about what you're going to do. This is a problem that I have, and I get hammered on every time I give an Ames page to somebody else. So they say, too much background, more details, every single time. So it's something I'm priming my brain to notice. And I think in this case, both Serene and Jamin picked up right away that the technical things that they wanted to know weren't there. And in a grant this short, you got no fat. It's tight. So you have to make it as tight as humanly possible. And you just have to assume that, like, I'm going to believe you that IPF pigs are important, right? You got to tell me why, but like in one sentence. And so that I think that would help address each of these individual concerns. And then the last piece that no one else has mentioned, so I will, is intra-hub collaboration. So no discussion has been made of intra-hub collaboration in the grant. I think it would be relatively straightforward to think about some intra-hub collaborations around this. There are cells that are being developed in the Witsit hub that might also be reasonable competitors to engraft into a bleo, where you could say, oh, we're going to use this cell and this cell. So if you structure the question the way Jamin said, maybe you can get cells from us, from our mice. There are a variety of different things you could think about with what the other hubs are doing that might make that piece a yes instead of a no.
Can I just add one last thing, Will? I think in the written reviews, one of the things that was a sort of common theme was the lack of preliminary data. And when you have a project that's, particularly if it's kind of a very high risk project, not been done before, and you state in the application that no one's made a pig, maybe cells from pigs or actually other large animals. So if you're going to propose to do that, reviewers not only want to know that it's feasible, but it's feasible in your own hands. So preliminary data can be a very great way to address that. Potentially in this type of application, you may not gotten that far to have preliminary data, but in a larger grant, it's it's an investment in you and your project. And so if you can't show that it's likely going to be doable and doable by you, do you have the resources? Do you have access to the cell lines that you might need, that kind of thing? Then they aren't going to want to give you the money. So preliminary data can be very helpful in convincing reviewers that you have the technology, you're capable of you have the resources, and you're capable of generating the model that and what's always tricky is how much of the grant you have to have done before you actually get the funding to do it.
Yeah, I mean, I think the key piece of preliminary data that both Serena and Jamin identified was the idea that you could grow these things and show us a picture of them growing in your lab doing something.
Yeah.
Right? Something.
Not one picture.
You don't have to show me definitive editor.
You
I mean,
clear
You don't have
but, you don't have to, right?
Yeah,
But like,
even as simple as just maintenance of these cells is sufficient.
Yeah, I agree. I agree with that.
Yeah, I was going to add that too, because the references and figures page, there's that like half a page of empty space where you can definitely take advantage of, even without even just a figure of the cells. And also if you want to make a schematic of the approach that you want to do, especially if you want to take on like media -1 experiments or list out specific, approaches that might not fit in your actual one-page text. I think, use whatever space you have there as well. And small thing to add on to what everyone else has said about the huge amount of background is your significance section, I think, bleeds into the approaches. So it'd be nice to separate.
Mm-hmm.
I know the first heading doesn't say approaches or methods, but I think it's useful to include that with anticipated results in some way or form as well.
All right. Anna, any more questions or comments from you?
No, actually, thank you so much for all the comments. I think now it's structurally will be easier to organize it and restructure even for the reviewers to read more in a good way. So thank you very much for all the feedback. Yeah.
Great. I mean, I agree with Dan completely. I would like to see this done. So I hope that you're successful in your resubmission. So as the one grant in the bunch, you've impressed the lung people. So keep going.
Thank you.
All right. We are actually more or less right on time, which is good.
Yeah, amazing.
So Danielle, you're up. I am also going to be your primary reviewer. So this is an interesting grant to review. And the reason it's an interesting grant to review is because the proposal is basically to purchase a piece of machinery that will enable the applicant to do some additional testing of a technology that's being developed as part of an ongoing project in their group. So essentially what the applicant proposes to do is to take induced pluripotent stem cells, engineer them towards cardiac-like cells, and then use those cardiomyocytes to generate grafts that can be used to eventually repair hearts of patients that have had injury, generally from something like a myocardial infarction. I think the argument is strong. It's a technology that needs to be developed. There are very significant limitations to regeneration of cardiomyocytes in vivo, and so there's a, I would say, a strong translational rationale. And I think that the technology that's being moved forward here is definitely an important translational research question. And so I would say an application of new technology to translational research is a clear yes in this grant. I think that's the strength of the grant. The challenges in the grant are related to the ways in which surrogate markers can be used to define potential in vivo outcomes for things like a cardiac grant. And so the positive argument that is brought forward here is that the best way to determine whether a graft will be successful in an animal model is to carefully understand its properties ex vivo. And this grant focuses on the ex vivo mechanical properties of the cardiomyocyte grants, which is an important question, and is a question that relates specifically to the type of tissue that the applicant is attempting to differentiate, because the mechanical properties of cardiomyocytes are a fundamental aspect of how they function and therefore are very important. So I think that rationale is very well described. And I think of the grants that we're reviewing today, this one is the best written to make the strongest argument for what is being done. I think that it has been nicely described what the central hypothesis, rationale, and long-term goals are here, and I think they make sense. The problem comes in being convinced that the testing that is being proposed is going to be a game changer for the technology itself. So the way that they propose to do this is to essentially buy a piece of equipment to look at different types of tensile strength on their cardiomyocyte grafts beyond what they've been able to generate. They've generated stress relaxation curves, which are, I think, convincing that the grafts have some value and that they replicate some important aspects of the in vivo situation that they're attempting to replicate. But I am not convinced that the piece of machinery that they're proposing to buy necessarily changes the fundamental understanding of what's going on with this graft. So it may be that it does, and I just have not seen the argument come through. But I think that's where the rubber hits the road for this grant. There's no inter-hub collaboration, which is a negative as well. And so while I think there's a strong translational research question, and the objective is clearly stated, it's not necessarily clear to me that new preliminary data will come from this, and there's no new collaboration. So we only get 2 out of 4 of the criteria as a yes, which then leaves me with trying to take the positives and negatives of this project and push them together. This is a common problem in grant review. I am a non-expert on the piece of machinery that the applicant proposes to use. And unfortunately, the argument has not been made well enough to convince me as a non-expert that this is going to change their fundamentals. You don't know 100% for sure if the people who are reviewing your grant are fully expert on each of the techniques that you're using. They theoretically should be experts on biology, and as a stem cell biologist after a fashion, I'm probably within the pool of people who could review a grant like this. At NHLBI, I might not be a primary or secondary reviewer in the real world, but I would certainly could sit on a translational panel like this that would be reasonable. And so I haven't gotten the full argument yet. So I gave the grant a 5 because I really like the idea, and I would like to know the things that the grant proposes to find out. But I was not able to convince myself that the approach of this piece of machinery would solve that set of problems. So either I think the question needs to be reframed around a less clearly technique-driven question, or more justification needs to be added in relationship to the precise ways in which this machine is a game changer for this technology, such that I then am saying, oh, they're going to spend this entire budget on an apparatus, but I'm convinced it's worth doing because this will work. And as Dan said, there are some things that are very high bars. I'm going to buy a machine and it's going to solve my problems is one of the highest bars in grant review, which is why it's very hard to try to find grants that let you buy an expensive piece of machinery, because I think review panels in the NIH and other funding agencies in general tend to be sort of dim that the problem will be solved by the new piece of machinery. And so that's my fundamental problem with the grant. But I like many other aspects of it, and I think if that could be finessed, there's probably a really important question here. And I think that the argument about the mechanical testing of this graft is really well done and needs to be expanded and focused on, because I think that's the core of what's really interesting here. Jamin, are you reviewer 2 with me on this one?
I'm not sure because I wasn't really given any review 1, 2, 3. So why not?
All right, do it.
Yeah. All right. So I think I agree overall with what Will said. So in terms of all the 3 grants that I've read, there are key phrases that I think any reviewer would look for. What is the central hypothesis? What is a long-term goal? As it's really nicely laid out. And but that being the case, I had difficulty finding what the question is. So for me, the hypothesis is not necessarily a question. Hypothesis is what you're going to test, but what is the exact biological question was something that was missing, which makes sense because it's pretty much the entire application aims to purchase an instrument. But for a grant application, what I would like to see in this particular, even if you're proposing to buy an instrument for the amount, is that what are you going to test on it? What are the, and by what I would mean, do you have an alternative to what you're going to test on this particular machine? Are you testing 2 or 3 different or 2 or 3 different materials? How are they going to compare with each other? And second, the fact that you have a new instrument, does the applicant know how to use the instrument? Is there a system in place near the applicant, either in the lab or in the hub or even somewhere else, where somebody has used this instrument and has shown that this is indeed correctly measures what they want to measure, and can actually train the applicant in a specific manner? I think there would be something would be really good. What I would think that, in fact, this might be something really useful as a visiting scholar application, in fact. I'm not proposing that change that into that or anything like that, but I think this would really help that if you had even just a letter from somebody who has used this instrument would kind of strengthen the confidence that I would have in this particular application. Overall, I think the section on approach is missing. Although it's described, it's an innovation and significance is kind of intermingled within the 2. And it's understandable because in the grant RFA title itself, the page doesn't say approach. So it makes sense that you probably miss it. But for any grant application, I would always imagine that an approach is the most central part, if not the central part. All the other things are bells and whistles, which will always break your grant, will never make it. So I think something along those lines. At what point in the differentiation protocol of your iPSCs are you going to put them onto this particular instrument? And I think I'm going to also be, just call myself out. The questions I'm asking could be completely useless questions because I'm not in the field. But that has to be clarified. Just a simple figure of what the proposal is would suffice to give a basic understanding to somebody who is a non-expert in the field to then comment on yeah, this makes sense. This is a good approach. Something along those lines. I think overall, I would give it a 5 as well. Actually, I don't know if that's what we'll give. I forgot already, but I would give it a 5. Primarily because I don't know what the question you're testing is. So.
And I would jump in as the third reviewer. I gave it an overall impact score of 4. And I really, it's the, plus or minus 1 essentially, but I totally don't have a whole lot to add beyond what you guys have just said. I really have the same, I think the underlying question of looking at the biomechanical properties of these graphs, I am totally convinced is really important. But the limitations of the grant are essentially what you guys have just said, so I don't need to repeat that. I think probably the way moving forward is, Will gave 2 options. I think probably the best way moving forward for the grant is try to rephrase it as a specific hypothesis, hypothesis-based question, a specific research question that you're going to ask surrounding the biomechanics of these graphs. And less as a sort of a application of a technology or tool that you're going to buy. And so I think like we've talked about, a lot of this is just framing and salesmanship as much as it is anything else. I think the biology that you're trying to understand, the biomechanics of these graphs, is really important and central to advancing the technology to therapeutic application. I think but probably, as we've all kind of alluded to, I think it becomes more, the way it's read is more of, I'm going to measure something and presenting it as I have a specific hypothesis, and here's the question that I'm going to answer using this tool, would be a more effective way of presenting the grant, which is just a different way of saying what you guys have both just said.
I wonder if there's, for example, an example you can give saying, look, people used to make thinner layers, but unless this particular hyper viscoso elastic properties were not met, like that would not work. Something of that nature, like as a very quick background, as like, if this is not tested, the thick layers would mean nothing, like simple, quick, something of that nature.
Thank you. That's very helpful. I really appreciate it.
Yeah, I mean, I think what comparing to what was written in your formal reviews as well, I actually think we came to a similar conclusion as what those reviews read to me, which is that everybody thinks this is a really cool thing, but that finding a way to describe the challenge that you're addressing in a way that is really hypothesis focused.
Okay.
Would and I think Jamin's question of could you see if someone else on one of the campuses of one of the other PCTC hubs has access to such a machine, and could you get a letter from them saying that you could go up there to try it out, or could you do a visiting scholar thing, or is there a way to generate some preliminary data that validates that buying the machine is worth the price? I think if you could address those 2 things, this goes from being a very challenging application to sell to a really high priority application, and then you could, get the thing you need and push forward on the testing you want to do to advance what I imagine is one of the important aims of your thesis. So
perfect. Thank you so much.
Will, Danielle, I wanted to ask what you thought of the clarity with which the translational aspect of the use of the technology was going to be. Did you feel, Danielle, like you said, look, once we have this technology going, it's going to help us select the exact graphs that we're going to use, and that's how this is going to translate into the application of future experimental work and ultimately clinical work?
So oh, go ahead. Sorry about that.
Oh, sorry. I thought the question was.
Nope. It was for you. It was for you.
So I might have not stated it as well as I would have liked, probably in the proposal itself, but yes, the idea behind it is that if we can mimic the cardiac niche really well in terms of cell population and things like the mechanical forces, for example, that B cells are exposed to, then we can select for the ideal fabrication method for these patches that we're making. And that includes whether it's simulation to make them more mature later on, all of these things. So it's a step that we need to go forward to make these patches basically optimal for not only therapeutic applications, but for diagnostic applications as well.
Yeah, so I think Michael makes a really key point, which is that is the central argument of the grant, and the fact that you feel like it might need to be improved, I think is an important thing to take forward for your revision. Because you implied that heavily, and I got the flavor of that, but looking at the range of reviews that you received, the people who were more convinced about the translational importance of this project liked it better than the people who were less certain as to what the translational impression was. And I think the one, lowest score that you got in your prior review really sort of says, great lab, important question, I don't get it. Is sort of the take home of what that review is, which I think is really along the lines of what Michael puts out. Jesse, Serene, did you guys, in looking at this one, do you have anything else beyond what we've shared?
For me,
Go ahead.
sorry, go on, Serene.
I was just gonna say, and Dan actually sent it to me. I didn't have like the CV, for example, or reference letter, so I don't know like if it was just a technical thing or I just didn't get it. So I couldn't even evaluate in some of these things, for example. And then the other thing that I would suggest is maybe like a gestalt of what you guys are saying is the organization of the presentation was a little bit like, there are paragraphs that I would bring a little bit earlier on, talk about your the elastic parameters a lot earlier, because then that information is buried in the significance, while this is like the main point. This is your title of your proposal. It took me like 3 minutes to get to reading that part in the third, at the end of the third paragraph, while I'm expecting that to be in your first aim, for example.
Yeah, I was just going to mention the same thing as Serene in terms of bringing up maybe significance to higher up on your page. I thought the innovation section wasn't really about innovation, so maybe that's more of a misunderstanding on my part. But at least to my understanding, layer by layer, has been done previously in tissue engineering. So it by itself, I don't think is something novel, but I think it'll be useful, especially in the figures, if you have, a prior sort of methodology compared to layer by layer, just to show the difference of how exciting this technology is for the cardiac field. I think would have been really much of a strength. A smaller note on the figures is I see that there are like significance bars on your viscosity measurements. And the first look, I was like, the legend says that they are mimicking native mouse cardiac tissue, but the significance bar threw me off, as in, like, do you see a difference between your iPS cell-derived cardiac tissue versus the mouse tissue? So it's something small like that kind of throws me off a little when I'm looking at your figures, that I feel like should be just a very clear message of, look, this is very similar to what's actually happening in vivo and primary tissue.
Yeah, I agree. The bar charts are always bad. Box of whiskers forever.
Okay.
Yeah, and just to reemphasize the point of the hypothesis, I think everyone, including Dan, also mentioned that it seems like a very goal-oriented proposal, which from an engineering standpoint makes a lot of sense. But for biologists, I think the hypothesis is really important. And the hypothesis I see in the current form of the proposal is that the layer by layer tissues will have similar mechanical properties as native tissues in terms of certain hyperviscoelastic measurements, which is kind of like and part of that hypothesis could be that based on the similarities between these properties, this makes such a method such a protocol for making the tissue very applicable for, I don't know, therapeutic objectives in the future. But to just, know the difference between the goal and the hypothesis and state the hypothesis more clearly, I think is very important in this grant.
All right, Danielle, any other questions or comments from your end?
I did have a question about just in the review sheet. I did notice that like 2 of the reviewers mentioned that something about the budget. They said it's not clear how the budget would be applied, basically, something like that.
Yeah, so I think that gets back. So I think the central problem that they had and the central problem that we have is that the explanation of what the machine you're proposing to buy will do in terms of advancing the translational objectives of this project is not well stated.
Okay.
And so one person sort of said that it was just a weakness of the grant was that you were going to buy a machine. But the other people were sort of like, it's not clear how the budget will work, which means why are they buying a machine?
Okay.
So I think that, we gave you a lot of sort of competing thoughts in this one. In the paragraph I'm going to write to you, I'm going to try to offer several of the suggestions of the various people in the call. It's possible that Rose may be able to share with you the recording link for this one as well, to be able to listen back to some of the comments that were made if that's helpful. Yours is the toughest nut to crack for a couple of reasons. The first one is it's the most heavily engineering focused. We don't have an expert in mechanical properties or cardiac muscle on the panel. Your question is technical related to the structural properties of cardiac muscle. But I think that what comes clearly through in the discussion of the others here is that we want to see a clear biological hypothesis of exactly what you'll learn. And I think you need to define what the relative added value for translation of a new piece of equipment is in a very clear way. And I think Jesse's suggestion of a schematic is dead on. That would help me personally. That would help me a lot. A good schematic here. Okay.
Perfect.
And sorry, just did I miss this? But was there a budget and justification of approaches page for this grant?
So guys, what the 3 of you received, there was not. What Dan and I received, there was. And Jamin also got it because this was his assigned grant, but he had to ask for it. So there was some sort of technical problem with getting the whole thing, but the budget justification, basically the letter and the budget justification basically say this is an important translational problem. We need this machine to solve it. And then the budget is buy machine, use machine. Seriously. So, and I think that was what it's okay because it's actually what you were proposing to do, right? I mean, it's
yeah.
The machine answers the question you have. The question's important. Everybody gets that, right? So, but I don't think it would have changed your concerns about the grant for either one of you guys, Serene or Jesse, because I think it emphasized that's the central argument that needs to be really brought out. But yeah, we didn't. There was back channel communication that you two didn't see. But I actually do want to say I am incredibly thankful to the 3 of you for joining us and for the hard work that you did in reviewing these grants. I think that your contribution to this made this dramatically better than the other ones that I've been a part of, because it gave us a diversity of reviewers and and comments that I hope are really going to help out the people who are applying. So I'm really appreciative that the three of you did it. And Jamin, you get an extra gold star because it was before dawn when you got up this morning. I would not have said yes to a 6 a.m. thing as a postdoc. I'll just tell you that right now. I don't want to say 6 a.m. things as a PI. Come on. So
that's why I didn't volunteer, but if I'm asked, I'm going to say function.
I thought postdoc was the bottom of,
yeah, I don't know. The only thing I get up at 6 a.m. for is ICU routes, so I can't really.
So I had I know, would it be okay to make some general comments?
Please, we got a half hour, so I was going to let people leave early, but let's summarize and whatever anybody else wants to talk about.
Well, in that case, I'm going to stop talking if you are going to let people leave early. I'm kidding.
Will, what did you just do?
The one general comment is that I think is part of, as I think Dan calls it, like selling yourself or the salesmanship. And I think it's absolutely right. It's part of the grantsmanship to understand what all you have available to you when you apply for when you write an application. An example would be that I think for Anna's grant, in her, I think, a support letter, there was something that was mentioned that as part of her postdoc project. So anytime you write something, the supporting letters are not the primary. So what I mean to say that is, just because it says in the supporting letter, it's not going to supersede what you didn't say in your primary application. The reverse is true. So what you could do is that, this is not just for Anna, but I think we're all in any grant, is that in your write-up, just refer to other sections, especially because you're always strapped for space. So as written by or as supported by this Dr. Schnook, this is what I'll be doing. And then you don't have to go in details because that letter is going to speak for you. But you have to refer to it. It's almost like you have to refer to as a reference in your grants to draw the reviewer's attention to that particular piece of addendum. Same thing, I think overall, I think every grant, everybody kind of mentioned that you have a lot of space in budget and justification or reference section, kind of minimize some of the other things and maximize the space to your advantage. Again, I'm just going to pick out examples. Is that for a one-page grant, 22 references might be an overkill. So you could potentially just cut down some of those references and then use that space for more figure, more details about the explanation. So I think it really is about which you can prioritize and which you can't. And a lot of it is about that. It's not black and white. It's really a gray area. I think that'll really help, not just this one application, but in overall how to improve the grantsmanship is really key to how you write anything. So there were a couple of just general comments that I had.
All right. Rose, any final words from you?
Yeah, just maybe two. One is, I think we did a little bit of talking about how reviewers will come to their final overall impact score, and then it's not just an average of the different sections. But I think that in general, I think Jamin pointed out that the approach section is always a really important section. But I would say the other section that really informs most reviewers' final score is the significance section. Because significance is taking, if you on face value that whatever you propose to do is going to work, how is it going to move the field? Is it going to have an impact on the field? And then your approach is obviously how you're going to show that, in fact, you are going to achieve the aims that you propose. But if it's not significant, if you can do something, but it's either, it's not going to actually move the field, either it's already, just an increment, really just an incremental increase in what we already know, or you're using a technology such that it's, again, it's not going to affect the field. So significance is really important. And so I think made the suggestion to Danielle for her application to move the significance section up, because you've got to convince reviewers right from the start. The significance is a short part of any application, but it should come across really crystal clear what it is, your hypothesis, what you're going to achieve, and what this is going to mean for the field. And it can be really good if it actually potentially has implications more broadly, potentially to other areas than your very specific area. So writing a good significance section to your grant can really help improve your overall score. But if you can't convince reviewers that what you're doing is actually significant, even if you have a great approach, it's not going to score well, in my experience. So those are probably the two areas you really have to work very hard on, I would say. But these were great applications, and I think everyone did a great job, and I'm really impressed with the level of for the junior reviewers, they're very sophisticated comments on these applications. Very appropriate.
I too wanted to thank everybody. I want to thank Will and Dan for their great leadership. I want to thank everybody on my team, without whom I would fall down. I want to thank the brave applicants who submitted their applications for discussion today. There are a lot of people who don't have that level of courage or ambition, and I want to thank our reviewers, our previous Jumpstart Award winners and guests for making this a study section that, fingers crossed, will result in successful awards down the road.
Yeah, just get your grants, guys, in September, and then you can come back next year and be panelists with us.
And then maybe I may not have to wake up at 6.
This is your additional reward. You should get up at 6 a.m. on the West Coast.
So much fun.
Cool.
And I will make sure that this recording gets sent out to everyone.
Wait, Will, can I see the poodle one more time?
Yeah, hold on. Ellie, you're being requested.
Oh, she's.
So she's, come on up. Up.
Oh, hello.
She's 16 months old, and she thinks this is the greatest thing that's ever happened.
You can bring her to the.
Dan knows
this. To the NICU.
She's been my constant companion for months, because I've been on and off of ICU service every couple weeks. So I'm like semi-quarantined from my family. And so this is my best friend. She's my only companion.
Oh no.
I'm basically stuck in this office. It's to the point that Dr. Whitsitt makes fun of me about how I'm always in the office when he sees me. It's in the basement of my house. And everybody's giving me a hard time, but I have a HEPA filter sitting right next to me, so it's the safest place in the house. It's a negative pressure room.
Oh my God.
These are the problems with being an adult ICU doctor right now, guys. Please wear your masks and stay safe, please.
Done. Considered done. All right, guys.
All right. Good to see everybody.
Thank you, everyone.
Thank you, Will and Dan.
Really good session.
Thank you again.
That was
Every-
great, guys.